Hello student friends. Today we are going to learn cell cycle in detail. I am Dr. Alka Inamdr in Alka's Botany channel. Please like, share and subscribe the channel. The cell cycle is a process in which a cell grows and divides to create a copy of itself. Some organisms reproduce through the cell cycle, and in complex multicellular organisms, the cell cycle is used to allow the organisms to grow and to replace cells as they grow worn out. The cell cycle consists of 3 phases of interphase and 1 phase of division phase. G1 phase, in which cell grow and synthesis. In S phase, that is a synthesis phase, DNA synthesis takes place, and gap 2, that is a G2 phase, in which preparation of division takes place. These 3, G1, S and G2 phase, comes under the interphase state of nucleus, whereas after completion of interphase, cell enters into the dividing phase, that is a M phase, which produces a mitosis and cytokinesis. In interphase of nucleus, which is composed of G1, S and G2 phase, where cell spends most of its time, that is near about 90% of its time. Often called it is as a resting phase, but cell is not at rest, cell is not dividing, but it is active. Gap 1 or G1, which is also called first growth phase. It is a period of rapid growth. New proteins and organelles are produced during this phase. Chromosomes are unwound, that is in chromatin material is there, preparing for DNA synthesis, that is it is a base for synthesis phase. It begins at the completion of mitosis and cytokinesis and lasts until the beginning of S phase. This phase is generally the longest of the 4 all cell cycle phases, and it is quite variable in length. During this phase, it chooses either to replicate its deoxyribonucleic acid, that is a DNA, or to exit the cell cycle and enter a quiescent state, that is G0 phase. G0 phase, in which cell leaves cell cycle. It can be temporary or permanent, but not necessarily dead. The best examples are neurons, which comes in the G0 phase. Second one, that is S phase, it is also called DNA synthesis phase. Cell makes a copy of its entire set of chromosomes, that is DNA. Replication of the chromosomes is restricted to one specific portion of an interphase, which is called as S phase or synthesis phase or DNA synthesis phase. During this phase, each chromosome replicates exactly once to form a pair of physically linked sister chromatids. And DNA is the genetic code. Genetic information is stored on chromosomes. The coded information on chromosomes is called DNA deoxyribonucleic acid. Last one phase of interphase is a gap 2 or G2, or it is also called second growth phase, which is a shortest part of the interphase. It follows after S phase, and some cells can exit the cell cycle from G2 phase just as they can from G1 phase. Completion of these phases is known as interphase. In G2 phase, cell grows larger in size in preparation for cell division. It produces organelles and structures needed for cell division. The best examples are centrioles and nucleolus are duplicated during G2 phase. M phase or cell division phase, it is a dividing process after interphase. Whenever interphase state of nucleus completed, then and then only cell enters into the M phase or division phase, that is mitosis and cytokinesis phase. In this division phase, all the cell's energy is devoted to the process of cell division. M phase is divided into mitosis and cytokinesis. This is a cell cycle G1, S and G2, mitosis and cytokinesis. This is a cell cycle in which interphase shows G1 phase, S phase and G2 phase. In G1 phase, there is a material, chromatin material is formed, centrioles are formed and chromosomes are replicated. In synthesis phase, that is S phase, proper chromosomes takes place. In G2 phase, all the enzymes and material which is required for the division phase is synthesized during G2 phase. And after that, it enters into the dividing phase, that is a mitosis. In mitosis, again there are 4 stages. First is a prophase, metaphase, anaphase, telophase. In the cell cycles of both unicellular and multicellular organisms, for each event in the cell cycle, there is a trigger or a signal reactions. These signals control the entire mechanism of cell cycle. The points at which signals are necessary are called checkpoints. The 2 important checkpoints are G1 and at the end of G2 phase. During G1, signal is released to enter the replication cycle of S phase, that is a synthesis phase, while at the end of G2 phase, signal is released to start the nuclear division, that is a dividing phase of mitosis. The first checkpoint is a cell growth checkpoint, which occurs towards the end of a growth phase, that is a G1 phase. It checks whether the cell is big enough and has made the proper proteins for the synthesis phase. If not, the cell goes through a resting period, that is a G0 phase, until it is ready to divide. The second checkpoint is a DNA synthesis checkpoint. It occurs during the synthesis phase. It checks whether DNA has been replicated correctly. If so, the cell continues on to the mitosis phase. And third checkpoint at the mitosis checkpoint occurs during the mitosis phase, and it checks whether mitosis is complete. If so, the cell divides and the cycle repeats again. Thank you very much.